Okay, we can't really have a section on endocrinology without including at least some of the pituitary hormones. I'm going to narrow this down though for us. We're going to focus on one posterior pituitary hormone and one anterior pituitary hormone. And I'll do the anterior pituitary hormone in a subsequent video. So we're going to focus on this one posterior pituitary hormone here. Antidiuretic hormone, vasopressin, those are both the same. That's the same hormone. It just has those two names. It actually is also called arginine vasopressin, just to make things more confusing. So antidiuretic hormone. So means it's against diuresis, meaning it will reduce urine output. It's an antidiuretic. If you took a diuretic, urine output increases. So this results in reduced urine output. And Dr. Sildorf will tell you the mechanism for that. It's actually not that complicated, but I don't want to steal his thunder. So another way of thinking of this, I like to think of this as the no-pee hormone, as a shortcut. It doesn't turn off urine output, but students seem to remember the antidiuretic hormone as the no-pee hormone. So that's a way to keep that straight. And we'll understand how this hormone is regulated, what its physiologic function is. But I want to add one thing about the anterior pituitary here, just while we're looking at this figure. There are a whole slew of anterior pituitary hormones. Like I said, I'm going to focus on ACTH. But what if I ask you a question? What is the second messenger for prolactin or FSH or growth hormone? All of these are cyclic AMP. So it wouldn't matter, right? I could say which of the following is cyclic AMP, and I would list a bunch of these. And as long as they were anterior pituitary hormones, you would pick all the above, right? They're all cyclic AMP. So I included that for people who are paying attention. And that might be a nice, easy exam question. But all right, enough of that. Back to ADH, or posterior pituitary hormone. So there are only two hormones from the posterior pituitary. There's ADH and oxytocin, and both of those are neuroendocrine. So neuroendocrine. Meaning they just come from a neuron and released their, what would be a neurotransmitter is released into a capillary system. And you can see a couple of cell populations here in green from the supraoptic nucleus and part of the paraventricular nucleus, at least on, part of the paraventricular nucleus. We'll get to that in a second. And you can follow those green axons down here right next to this capillary system in the posterior pituitary. So this is released just like a neuropeptide would, a neurotransmitter, but it diffuses into the blood and ultimately out to its target tissues, right? Whichever receptors have ADH receptors on them would respond to this hormone. Now, a little bit about the nomenclature. Supraoptic nuclei. Look at this optic chiasm right here. Well, that's on top of the supraoptic chiasm, supraoptic nuclei, and there's one on the left and one on the right, and that's why it's plural. So that's fairly straightforward. Paraventricular nuclei. Well, these are on either side of the third ventricle. So third ventricle. And plural again because there's one on the left side, one on the right side. Now this magnocellular part. There are multiple cell types in the paraventricular nucleus. Two major populations that we're interested in. Magno just means large. So the soma on these cells are large. And just as a preview for next time, parvocellular neurons, that just means small. So these are small cells. Magno large, parvo small. And that morphology tells us a lot about what they synthesize and secrete. Again, these magnocellular neurons are going to be what will be secreting ADH for us. So we want to understand how it's regulated a little bit. I'll give you a second to get over your panic over this complicated figure here. We don't have to know everything on here. Just some of the basics. And I put boxes around the important bits. I do have a little inset here with some real anatomy. This is actually from a rat. But the anatomy and functionality is very similar to what we see in humans. So it's actually a pretty good model system for that. So let me start up there with that figure because that's the colorful part and it's probably what's drawing your attention right now. And I'm going to zoom in just a little bit so we can see these cells a little bit better. See this little inset here? There's some green cells and some red cells. Well, the green cells here, these are the ones that are going to be secreting. This is where ADH is going to come from, the green cells. And there is overlap with the oxytocin cells. That's the red one there. But we don't care about that for now. But if you look at this, we have this nice little kind of a ball of cells on either side of this. The paraventricular nucleus overall is kind of like this. So it's, I think of it as kind of a butterfly shape. Some people say it's a heart shape. And those parvocellular neurons are in here. But we'll get to those next time. But we're interested in these magnocellular neurons. So the magnocellular neurons are going to send their axons down through the infundibulum here to the posterior pituitary. So this is how ADH will be released. The supraoptic nucleus over here. Here's the optic chiasm. They're showing you the optic chiasm down here. This is our supraoptic nucleus. Also sends its axons down to the infundibulum here, and ultimately to the posterior pituitary. So that's where our vasopressin or our antidiuretic hormone is coming from. So I remind us again, these are the magnocellular neurons in the paraventricular nucleus of the hypothalamus. So it's kind of a mouthful. But the paraventricular nucleus is this whole structure. Magnocellular neurons are within this paraventricular nucleus. And the paraventricular nucleus secretes several things. We aren't worried about oxytocin. I have VP here for vasopressin. Again, that's just another name for VP is equal to ADH. Same thing. Unfortunately, let me tell you another name for vasopressin is AVP for arginine vasopressin. And usually only researchers use that full name for it. And that's just to contrast it with another type of vasopressin found in other species that have a lysine there instead of an arginine. But vasopressin, ADH, arginine, vasopressin, all those mean the same thing. All right, so now we know where the vasopressin is coming from, or ADH is coming from. Supraoptic nucleus and paraventricular nucleus. And now we want to understand a little bit about how it's regulated. And so this is involved in urine output, so you'd expect it to be involved in water balance, and it is. So antidiuresis. So you can imagine when you might want to secrete this, right? If you're dehydrated, you want to hold on to more water. But we'll walk through examples with this so that we understand it. There's a little structure here right at the most anterior part of the hypothalamus. As a matter of fact, it's the final lamina of the hypothalamus. That's where this lamina terminalis business comes from. And it's the organum vasculosum. There's a lot of blood vessels there. Lamina terminalis. And usually we just shorten this to just organum vasculosum. And this is where we find our Osmo receptors. So they're going to respond to changes in osmolarity. Ultimately plasma osmolarity, but of course that has to be ECF changes, extracellular fluid changes, and ultimately CSF changes. Because our detector here, our receptor for the system, is in the central nervous system. So before we get into this in any kind of complex way, or before we make this complicated, let's think about the overall logic here, right? So this is going to be involved in water regulation, and you're going to get more of this in the renal system, I'm sure. But the overall logic, well, if you're dehydrated, then plasma osmolarity, so osmolarity, will increase. And if that's not obvious to you, think about it this way. Let's say we had a little beaker over here, and there's some water in there. There would also be, we're going to make this like blood, so we're going to have some plasma proteins. We'll have some salt. Well, if we're losing water through perspiration, or if it's this beaker, just evaporation, well, that water level goes down. But we didn't lose any of these osmotically active compounds in here. What we did is just increase the concentration. But another way, we increase the osmolarity. It's just another way of measuring concentration of osmotically active compounds. And our Osmo receptors can detect that. And that's how we can detect dehydration through this system. All right, so if plasma osmolarity goes up, that means you can also think of this as blood is more concentrated. And your hematocrit could go up to reflect that. Well, what do we want to do? Let's put some more water back in here. Let's take that water that evaporated and return it to dilute this back down. The other thing you want to do is try to stop this water loss. And that's really how this system works. So add more water in. So what we want here is more, I'm going to just say H2O in, less out. That's how the system works. I know it looks complex, but that's really what it is. And whenever you see a really complicated figure or flow diagram, think about the logic, right? The regulatory systems. Well, we have a physiologic range for hydration. And we have multiple systems to detect that, and we will have responses according to how far out of the physiologic range we are and in which direction. And that's really all regulation is. And that's why I started out with those general ideas, because it applies to specific systems. So, well, let's just walk through what's really going to happen here with the system now that we know the logic. So, let's say you are dehydrated, right? So we have the first leg of this. So, dehydration leads to increased, I'm going to say, plasma osmolarity. Which ultimately is going to change ECF osmolarity. And ultimately CSF. Right, so this is increasing all of these guys. All we did was took the steps from blood all the way up to where our detectors are next to the CSF. So that's going to activate our Osmo receptors. And by activate, I mean the output's there. Remember, we always have some basal activity. What we just did is we increased the frequency of action potentials coming out of that system. And this is going to send inputs to the supraoptic nucleus. I'm just going to abbreviate it, and also activity to the, I'm going to say magnocellular, just to shorten it here, BVN. what happens with these guys? I'm going to start going down, so I'm going to need some room over there in a moment. Secrete ADH. What's our ADH do? It's going to reduce urine output. All right, so there's water out. We're going to stop losing the water. So we're going to try to retain the water we have. How are we going to get more water back into this system? Well, in addition to this, so the Osmo receptors are activated. I'm going to use a different color to indicate this. We're also going to activate lateral nuclei, the hypothalamus. I know, I haven't talked about that yet, have I? Let me show you what that does. Oop, come on, there we go. So I have lateral hypothalamic area. There are multiple nuclei in here, so I'm just calling this lateral hypothalamic nuclei. And there's this ventromedial region. There's only a couple of things we have to pay attention to. Thirst and satiety for this. If we activate the lateral side, you get thirsty. If we activate the medial side, you are sated. You don't want any, you don't need to drink anything. And we can also think of these as being, as a form of reciprocal inhibition. It's a little bit of an oversimplification, but it's okay for us. And all that means is if this is activated, this is inhibited, and vice versa. If we activate the ventromedial, we'll inhibit the lateral. And logically, you aren't going to be thirsty and sated at the same time, right? So these are going to work opposite of each other. So what we just did was we activate the lateral hypothalamus. Look at that, you got thirsty. That doesn't make sense. You're dehydrated, you're going to be thirsty. So thirsty by activating those Osmo receptors. So we have two aspects of this. One, we're going to try to, assuming you have access to water, you will add more fluid to the system. Here we're reducing fluid loss. Again, restoring back to normal. And if we are able to do this, right, so we can actually just go right back to the plasma osmolarity here. If we dilute the blood back down a little bit, we reduce the stimulus here. And this will keep going until we get back into the physiologic range and we're no longer activating those Osmo receptors. I guess I could just make it even simpler and just go right to the Osmo receptors there, right? By through this mechanism, by rediluting the blood back down again. That's essentially it. Now, hydration state is actually fairly important. And we have more than one set of inputs to this. So I do want to bring in cardiovascular influences on ADH production as well. So right up here, we have baroreceptors. Go ahead and use a different color here that shows up a little better. So there are baroreceptors. And baro just means pressure. So it's a form of receptor that responds to pressure. So this is a pressure receptor. But it responds to pressure through stretch. So if you think of this as a tube, and we've got these little sensors on here, these are going to be from dendrites of neurons. Well, if this expands, so this diameter increases, that's going to stretch. So we get stretch. So that's how we detect pressure, is really by detecting stretch right here in this coronary sinus, or the carotid sinus. So that is going to detect blood flow up to the brain. So it's very strategically placed too. If we get stretch, we'll activate this system. All right. Now, let me just walk you through how that responds if we get stretch, and then we'll look at what happens in a case where we have potentially dehydration or a loss of blood volume. So pressure in the system is what's going to lead to the stretch. So what will activate this is an increase in mean arterial pressure. So if our arterial pressure increases, we're going to get stretch. Just like I showed you a second ago. Well, that increases the firing rate, or our action potentials coming out of the neurons that come off of this. So firing rate increases. So far, so good, I hope. This inputs into an inhibitory center. I'm just going to say inhibitory center, just because I don't want to get into a lot more of the anatomy here. So if we increase this firing, what we're doing really is we're going to increase inhibition. And that's actually a fairly common method of regulation in the nervous system. And again, I wanted to give the example just so you didn't think this is always stimulation, response. Here we have stimulation to inhibition, so it's like we're putting our foot on the brake. And the response that comes out of this through a couple of cardiovascular centers is going to be a reduction in heart rate and also a reduction in vascular resistance. I'm just going to do, I'll go ahead and write it out. And also, it's going to reduce ADH. That one might take a second to think about. Remember the other name for ADH? I also called it Vasopressin, didn't I? So, Vasopressin. Vasopressin causes vasoconstriction. And that's where that name came from. We actually learned about its effect on the vasculature before we learned about the antidiuretic effects. So vasopressin was really what ADH was called first. If it's elevated, we get vasoconstriction. But if it's lower, we get vasodilation. So that enhances this reduction in vascular resistance. So that's going to help lower our blood pressure. So that's ultimately going to help us correct this system, just a little negative feedback loop here. In case you don't remember, right? Times vascular resistance. Heart rate is a part of our heart rate times stroke volume gives us cardiac output. So we're working to reduce these things and bring arterial pressure back down. So that's the vascular side of this. And that's what happens when this system is overloaded. Well, the opposite happens if mean arterial pressure is low, which is what you would expect if you're dehydrated, right? So what if we had lowered mean arterial pressure? So less stretch, reduce the firing rate, that means less inhibition through that inhibitory center. So I'm just going to jump to the end result here. Less inhibition. Well, that's going to give us the opposite response, isn't it? So we're going to get increased release of ADH, increased heart rate, and increased vascular resistance. Just shortening it here. Now, these two are mediated by the sympathetic nervous system. This is ultimately going to be neural inputs to an endocrine system, so we're going to get an endocrine response too. Elevating ADH, remember, it has a vasopressive effect will contribute to this, but also think about fluid volume. So if we increase ADH, this is going to result in less urine output. What else happens? Same thing as if we stimulate it through the Osmo receptors. You get thirsty. And you've probably seen this in movies and didn't recognize it. If somebody's bled a lot, their blood pressure goes down, and usually they're complaining about being thirsty. Well, that's through this ADH mechanism. And also through activating, I guess I should say, this is the lateral hypothalamus. That's the direct mechanism. Just so you know, ADH also, well, I'm not going to get into that complicated part of this. All right, so these two are working together. So if we're dehydrated, we have the dry or less inhibition coming in, effectively stimulation by removing that inhibition on ADH, in addition to the cardiovascular effects. That would also feed right into what happens with these Osmo receptors being activated with dehydration. And so these two systems work together from different points of view. One is blood volume, and pressure. It's technically pressure because that's what the sensor is detecting. And this system is based on osmolarity. So we can artificially activate these receptors and ask you what the output would be. So let's say, for example, I won't get into diabetes very much because I know Dr. Sildorf loves to do this for metabolism and a lot of things, and I don't want to steal his thunder. But diabetes. And this is mellitus, so this is going to be the type that involves sugar, blood sugar. So let's say we have elevated blood glucose. Caused by this diabetes. Well, this is osmotically active, so this is going to cause an increase in plasma osmolarity. Well, with an increase in plasma osmolarity, we're going to activate our Osmo receptors. So all of these responses are going to take place by activating the Osmo receptors. So ADH release, that's one thing that's going to happen. Thirsty, by that direct input to the lateral hypothalamus by activating the Osmo receptors. So people with diabetes tend to be thirsty, and they have high urine output though. What? The mechanism for this, we will get ADH secretion to try to counteract the urine production. But Dr. Sildorf will tell you about something called osmotic, or actually he'll probably call it, let's see, drag. And this is actually why you see an increase in urine output. In spite of the fact that we just activated those Osmo receptors. Again, I don't want to teach renal, but he'll tell you about how that works. That's going to interfere with reabsorption of fluid, and ultimately that fluid will pass on through. And it will include glucose that passes through. So there's a lot of things we have to get into to understand that in renal. But my point was we could activate the Osmo receptors through another means. You don't have to be dehydrated. What if I put an electrode in here and stimulated that thing? You would get these responses. So again, think about this mechanistically. It's always good to understand the logic, but make sure you take it to the next step and understand the mechanisms too, because I could stimulate a baroreceptor and ask you what's the effect on ADH levels, right? You know from this, if we activate the baroreceptors, that ADH levels would go down, right? So just understanding these relationships between the mechanisms here will take you a long way toward answering any kind of question I can throw at you, because it's all based on physics, stimulus response, and that's really it. If I give you anything that does not fit with how the system works, then it's a bad question, and I would just throw it out. And that's actually enough for the posterior pituitary. I think we've got a reasonable understanding of how ADH is regulated. And that's really all we needed to do for this one. And I'll do the anterior pituitary hormone and ACTH in a subsequent video.